Recent research offers compelling evidence that food, particularly homegrown produce, represents a significantly underestimated exposure pathway for toxic, forever chemicals, specifically per- and polyfluoroalkyl substances, PFAS. A new study underscores this concern, revealing alarmingly high levels of PFAS in produce cultivated in home gardens located near a Chemours PFAS manufacturing plant in Fayetteville, North Carolina. This finding strengthens the growing body of evidence suggesting that food, especially when grown in proximity to industrial polluters, serves as a crucial, yet often overlooked, vector for PFAS contamination. The study, published in a peer-reviewed journal, analyzed 53 samples of produce harvested from 5 different home gardens within a few miles radius of the Chemours plant over a period spanning from 2013 to 2019. Remarkably, every single. This widespread contamination strongly implicates airborne emissions as a primary source of PFAS pollution, a hypothesis supported by an increasing number of independent research studies. The prevailing understanding of PFAS pollution has historically focused on other pathways, such as contaminated water sources, leaving the significant contribution of air emissions substantially undervalued. This study directly challenges that assumption, providing concrete evidence linking atmospheric dispersion from industrial facilities to pervasive PFAS contamination in the local food chain. The implications of this research are profound, particularly when contrasted with the Food and Drug Administration's FDA approach to PFAS monitoring in food. The FDA conducts annual testing for PFAS in food products, yet these tests rarely detect the chemicals. This discrepancy has prompted considerable criticism from independent researchers who contend that the FDA's methodology is inherently flawed, deliberately designed to underreport the extent of PFAS contamination in the food supply. Critics point to the FDA's unduly high minimum detection level threshold as a primary source of this underreporting. This artificially high threshold allows the FDA to claim non-detection, even when potentially hazardous levels of PFAS are present in food samples, effectively masking the true scale of the problem and minimizing public awareness of the risk. An example of this methodology's limitations can be found in the FDA's 2018 testing of 108 produce samples near the Chemours plant. While initially detecting PFAS in a significant number of samples, the FDA subsequently altered its methodology, resulting in a revised dataset that reported only 36 contaminated products. This significant reduction in reported contamination, coupled with the agency's downplaying of the associated health risks, has raised serious concerns about transparency and the reliability of the FDA's findings regarding PFAS in food. The discrepancies between the initial findings and the revised data, along with the shift in the agency's communication regarding the risks, fuels skepticism regarding the accuracy and objectivity of the FDA's monitoring efforts. The public's trust is understandably eroded by such inconsistencies and the perceived downplaying of a serious public health concern. Further bolstering the concerns raised by the study's findings are the results of independent air quality testing conduct. Commissioned by The Guardian, this independent assessment revealed PFAS levels in the air surrounding the Chemours plant that significantly exceed the values reported by both regulatory agencies and Chemours itself. This discrepancy highlights a potential systematic underestimation of PFAS air emissions and the associated environmental and public health risks. The disparity between independent findings and official data underscores the necessity for more rigorous and transparent monitoring, coupled with a reassessment of the current regulatory frameworks governing PFAS emissions and food safety. The implications of this persistent disparity extend far beyond simply questioning the accuracy of data. It highlights a potential systematic failure to adequately protect public health and the environment from the insidious effects of these persistent pollutants. The cumulative evidence strongly suggests that a comprehensive reassessment of PFAS exposure routes is urgently needed. Food, particularly locally grown produce near industrial sources of PFAS pollution, needs to be recognized as a primary and potentially underestimated vector of exposure to these dangerous chemicals. The discrepancies between official data and independent research, coupled with the alarming findings of recent studies, underscore the critical need for more robust regulatory oversight, improved monitoring methodologies, and increased transparency to effectively address this pervasive public health threat. The health and safety of communities living near PFAS manufacturing facilities, and indeed the broader population consuming potentially contaminated food, demand immediate. This necessitates a fundamental shift towards a more proactive and precautionary approach to PFAS regulation, safeguarding both the environment and public health from the long-term consequences of these persistent pollutants.